Okay, so we're gonna start now. So we're gonna start one of the keynotes of RIIAA for this year. I would like to present Liset M. de la Prida. She is a leader of a Laboratorio de Circuitos Neuronales del Instituto Cajal in Madrid, in España. And she has a talk about AI meets the brain, understanding memory with machine learning tools. And just before we start, I'm going to be moderating this keynote. My name is Benjamin Sanchez, and we have in support Jennifer Enciso, which is going to be in chat and helping us out. And just to start, we're gonna have a Menti online website that you can visit, and you can put a code in, and there we're gonna have any questions. So any questions that you have during the talk, please put them there, and you can vote for questions there. We will also have a possibility to have live questions. But so so how about we start, and I'll pass it on to Liset.
Thank you. So can you see my screen? Can you see the mouse?
Yeah, good. We can see your screen and mouse.
Okay, great. So I would like to thank the organizers and especially Miguel Angel Núñez who invite me and for the opportunity to talk here. This is a very interesting event, which is focusing artificial intelligence, and I am a neuroscientist. So my main focus is to try understanding how biological brains implement cognitive operations. So I assume that what I can tell you is basically related with how we can use machine learning tools to try to understand how the brain works. But hopefully, I also will try to, if you follow me through the talk, I hope you can get inspired by the solutions that the brain has for learning and memory, and maybe to try to think that artificial intelligence still need to understand or to learn more from the brain itself, so that the tandem between artificial intelligence and the brain is and should be more circular than ever. Machine learning is an application of artificial intelligence in which we give computers data, large data set, and we ask them to solve a particular problem. We can use simple architectures like recurrent neural networks or feed-forward neural networks, which actually are inspired in biological networks. So this solution works pretty well for simple pattern recognition tasks, like, for instance, training a multi-layer networks to recognize digits, handwritten digits, numbers, using some kind of supervised learning. This very same type of artificial networks can be trained to see, to hear, to perceive the world, but we all need to feed them with large training data sets. However, some other tasks are rather complex for artificial intelligence to learn. A now well-known example is the board game Go, which is considered hard to learn by artificial neural networks. And but recently, DeepMind launched AlphaGo, which successfully beat professional players of the game. The key point about AlphaGo was the strategy. So as before, AlphaGo used a network that was initially trained to mimic human play using a data set of millions of moves. But once it had reached a certain level of performance, it was training by self-playing with reinforced learning. Demis Hassabis, who is the founder of DeepMind, as you all know, was actually a neuroscientist. He had this experience from before, and he knew that the brain self-plays to learn. But the way brain self-play is by repeating activity patterns which are activated by experience. And there is one particular region in the brain which do this job very well, and from which we know the most how it is involved in this reactivation self-play game, which is the hippocampus. The hippocampus is critical for learning and memory. So while artificial neural networks can solve simple problems by mimicking the way we see scenes, they can classify pictures or images, the way in which the visual cortex operates from the input layers to hidden layers, it seems that we can also get advantage of learning how the brain actually learn by self-playing, using the information that we record during experience and reactivate this information in a way that helps to simulate, also to repeat and to somehow improve and consolidate learning. So why these neural networks can solve these simple problems, machine learning still requires additional training policies, algorithms to interpret, to generalize, and to operate. So the solution somehow is hidden in the brain. But the hippocampus machine is so challenging that we are still trying to understand how it operates. As you probably know, in 2014, the Nobel Prize was awarded to the discoverers of place cells and grid cells. Place cells were discovered in rodents when you record and you isolate activity from a single neuron in the hippocampus of a rat, which is moving around in a room, and you see the cell activated at a specific location in the room. This is called the place field of that cells, and it is said that this cell represents space. There are other neurons which represent has place fields in other location of a space, and so you can cover the whole space with the representation of these place fields in the C1 region of the hippocampus. The entorhinal cortex, which is the input layer of the hippocampus, has other cell types, which are grid cells, and grid cells, they fire in a grid, which tesselate the space. The entorhinal cortex and the hippocampus, they together with other associational cortex, they form the temporal lobe. And in the temporal lobe, there are other cell types that also code for other aspects of space, like border cells, which they are activated near the borders, like near the walls or near the objects which are in the middle of the room, or head direction cells, which fire maximally or are activated maximally when we are looking at a specific direction. The way in which the hippocampus machine builds these neuron representations is still unclear, but they presumably reflect the building blocks of a higher cognitive function, which is episodic memory. Episodic memory is the ability to relate what happened where and when, and this is a major cognitive ability required to navigate, to live, and to solve the everyday problems. Actually, in the human temporal lobe, there are more elaborated cells, such as concept cells, which they are activated when you present images of a particular person, like Oprah Winfrey cells that they fire when you see the for instance, different pictures of her, or the name of her, or you can hear the name of her. But this very same cell, they don't fire at all when you see other characters like Luke Skywalker or Darth Vader. There is a fully orthogonal representation between the different characters. So this is how hippocampus machine somehow represent information and can represent complex aspect of information. But the best trick of the hippocampus machine is at the operational mode. When the animal run around the space and they go from instant from one place to another, they go through the place fields of different place cells. And therefore, the activation of the different neurons in the rat hippocampus is sequential. And this sequence, this internal neuronal sequence, represents the sequence of experience that the animal or the subject is experiencing. And this is called the acquisition mode, and is somehow driven by the interaction between two main brain rhythms, which are theta and gamma. Theta is an oscillation between 4 and 12 hertz that you record in rodents when they move around. You also record theta in the human hippocampus with a different expression depending on whether it's more anterior or posterior. Gamma is 4 hertz oscillation, which are actually nested within the theta cycles. And presumably during this theta gamma oscillation, when the animal is moving through the sequence of cells that fire at the different locations that the animals are experiencing, are activated in a sequence. Later, when the rat is get to sleep or is immobile, the very same sequences are reactivated again. And this is a kind of repetition mode. And this is going on when in the hippocampus you record a different type of events, which is called sharp wave ripples. Sharp wave ripples are high frequency oscillations occurring faster than 100 hertz. And during these very brief oscillations, which are very short-lived, they are no longer than 40, 100 milliseconds. As I said, you have a reactivation of the same sequences that were experienced before. So the hippocampus inspired the solutions used by AlphaGo to complement supervised learning by using self-learning by repetition. But the point is that in trying to understand how this biological network implement the function, we need to understand the complexities of the biological machine. And this function is determined by the intrinsic properties of a diversity of neuronal types and the connectivity pattern that wire them specifically. For instance, grid cells from the entorhinal cortex, they could be either stellate cells or pyramidal cells. They express different genes and different proteins, like, for instance, reelin in stellate cells, or these other type of proteins in principal cells. They both exhibit grid cell activity, but they project differently into the hippocampus. Stellate cells, they project to the granular cell layers of the dentate gyrus, which is this structure in the hippocampus. And these granular cell layers, they then activate the CA3 pyramidal cells, which are wired as a recurrent neural networks. The axons from CA3 pyramidal cells project to CA1, which is another region of the hippocampus, according to a feed-forward network architecture. And the CA1 is the region where place cells were initially described by O'Keefe. But we have learned that how stable these place fields are in CA1 cells, according to, in response to external influences, strongly rely on whether the cells are located more deep or more superficial in the layers, because that determines the type of genes that they express, the intrinsic properties that they have, and the connectivity that they have. Like, for instance, how much they receive from the intrahippocampal CA3 and the CA2 region, which is another region involved in social memory, and from the entorhinal cortex. So this is so complex that just trying to explain all this to you will make us to lose the focus. And I don't really want to deliver a very complex talk about all these issues. I just want to try to discuss with you how we can learn from the beauty of the biological neural networks. And in doing so, I will try to simplify the questions and to concentrate in understanding how these wet neural networks could operate. These very simple CA1 networks of pyramidal cells and other cell types that I will explain along the talk, how they operate during ripples, during replay, and how that could explain the diversity of replay. Now, the more you look at the delicate assembly of the different neuronal types, the more challenging is to understand how nature has sculpted this solution along millions of years of evolution. So if you think that artificial intelligence takes long to learn, think how long it was for nature to optimize our brain. Here is where artificial intelligence and human intelligence should come together to try to solve the challenge. So in my talk, I will show you how we are using some machine learning tools to dissect hippocampal operation, in particular doing ripples and chart wave ripples. And to make it simple, I will only focusing how we are using some dimensionality reduction techniques and manifold learning to try to extract the dynamics and the organization of this, the variability of ripple and replay. And we will discuss, hopefully provide potential inspiration for the next generation artificial neural networks. So that in a way, we need to think that by understanding memory and learning, using these machine learning tools, probably we can close the loop to get artificial intelligence applied to try to understand the brain, and at the same time to modify artificial intelligence routines and algorithms inspired by these brain solutions to that. So, but to understand the many dimensions of hippocampal self-play, we need to start with the simplest. And the simplest is the chart wave ripples. Chart wave ripples are brainwave oscillatory phenomena, electromagnetic electroencephalographic waves that can be recorded with intracranial electrodes that we implant in the rat brains. And they consist of a high frequency oscillation that can be recorded near the layer where the cell bodies of the pyramidal cells are located, synchronous and together with a chart wave that is recorded near the regions where the apical dendrites of the pyramidal cells are. Chart wave ripples are the most synchronous physiological pattern of the brain that we know so far. This is because during chart wave ripples, we know that about 30% of hippocampal neurons are activated together during that short period of time. During a sleep, you have thousands of these reactivating patterns that are somehow reactivating a constellation of neuronal sequences, so that you can imagine that all experiences and the sequences of experience that you are getting through this conference, the way you get into the room, other memories that you have, and somehow establish the association, they are all replayed along these different events that occur in your brain. During sleep, this is typically associated with plasticity phenomena that occur both to depress and to potentiate synaptic wave. We still ignore when one or the other operates. So there are still many open questions regarding chart wave ripples and the way replay is performed. So, but how does it work? How ripples emerge and how they replace sequences? What are the ripple building blocks? Ripples reflect instances of a basic circuit motif, which is basically defined by the connectivity between a pyramidal cell and an interneuron. Pyramidal cells are excitatory cells. They activate other cells they connect with. Interneurons are inhibitory cells. They inhibit activity of the other cells they connect with. When you discharge a pyramidal cells, you make them fire. that recruit an interneuron. And there is a kind of interneuron which innervates very densely the perisomatic, the cell body of all pyramidal cells. In this drawing, you see each of these buttons that you can see here are synaptic contacts made by these interneurons of different pyramidal cells here in this region of the hippocampus. And that generates a positive waves which can be recorded extracellularly as a single event, which reflect the inhibition, the desynaptic inhibition exerted by this interneuron over a group of pyramidal cells. Now, if a pyramidal cell and an interneuron, they target the same other cells in the vicinity of the neighborhood where they are seated, that generates a sequence of activity. Basically, when pyramidal cells activate the interneurons deliver the inhibitory neurotransmitter, and you have a very short window for other pyramidal cells to fire before you recorded the positive wave of the inhibitory action of the interneurons. So that when you have this change across the network, you have a sequence of events, and that sequence of events reflect somehow the sequence of the ripple and the sequence of activity running through the network. So the pyramidal cell interneuron is a basic local microcircuit motif that underlines ripples, and the different neuronal sequences that emerge, because of they are somehow related with the way in which the different interneurons and pyramidal cells are activated, should imprint a unique extracellular signature. For instance, here you see electrode from number 1 to number 8, which were placed along the cell layer. And as you see, that the shape of the ripple that you record extracellularly is reflecting how close they are to the different components of the sequence. So the circuit architecture also matters, because that will determine how excitation and inhibition is shaped. And for instance, we know many microcircuit motifs that are actually being shown to have a specific computational role and to play a particular function. For instance, simple feed-forward inhibition between pyramidal cell and the interneurons allows for input normalization, and they help to increase the dynamic range. This inhibition, for instance, is quite useful for associative learning by gating the inputs. Recurrent excitation is well known as these recurrent neural networks. They have a robust feature representation. They are very useful for pattern completion, whereas lateral inhibition underlie competition for representation, and they are good for contrast enhancement of pattern selection, pattern separation. So somehow this the different microcircuit motifs should be activated during ripples, during different events, by reflecting the sequence of different sequences that occur, and somehow that should imprint a different signature at the level of the extracellular signal. We suspect actually that that will be the key to try to understand the many forms of self-play that the biological hippocampus, has. Indeed, you can see this variability emerging when you record ripples from a particular location in the brain. For instance, here you have examples of 3 examples of ripples recording in the very same electrode implanted into the hippocampus, near the cell body layers where you can record the ripples, and near the dendrite layers where you can record the sharp wave. And as you can see, if you compare the different events, you can see different waveforms, different shapes, different amplitudes of the sharp waves, and different spectral components, different spectral organization, from very slow frequency component to very fast frequency components. So if we focus on frequency, for instance, we can see that we can move from events with low somehow frequency components to very large, so that if we pick up the maximal frequency, spectral frequency, and we represent that in a distribution, we see that actually there is a distribution from slower to fast frequencies. And actually, the distribution of the ripple frequency correlates somehow with the amplitude of the sharp wave. So is dot here is a sharp wave event, the amplitude and the frequency recorded in a given location during slow wave sleep. And so you can see that the larger sharp waves are associated with the faster ripples, and the slower somehow could be associated with the slower ripples, but somehow there are some variability here. So can we understand this variability and extract information from this variability, and how can we use that to relate and to better understand how the hippocampus is organized? If each event here reactivates a specific ensemble and a microcircuit motif, there should be something unique in the sharp wave ripple waveform. And we should be able to extract that information somehow by looking at the event by event variability. So the idea is that if we record from a large number of events from a particular regions, and we sample this event at a high sampling rate, say 30 kilohertz, we can actually build a high dimensional representation of the different waveforms by using the different set points in a high dimensional space. So in particular, we can go up to a 1,500 dimensional space, which is basically what represents sampling 30 kilohertz ripples during a short time window. And the access in this high dimensional space will be the temporal latent variables. So the idea is that we can collect many events during a session and go to this high dimensional space where each event is a dot. So similar events will tend to group together along the different high dimensional axes. Like, for instance, events that tend to occur at the beginning with the frequency components at the beginning will go more towards the earlier axis as compared with events that are coming later, which they will mostly distribute to the other axis. And then you can try to apply some dimensionality reduction techniques, like the most simple one, which could be the principal components, to try extracting some of the topological organization in the original space. And by mapping at how the original events project the different components from the PCA, we can get information of how much of the variance can be explained differently. Like, for instance, the first component, which like explain 20% of the variance, is basically reflecting the maximal amplitude of the event, which is related with the sharp wave, as we said before. But then what you see is that this is telling us that we may be losing some information, because we know that there is just minor difference between the other components after the first component, which only explain the peak of the event. And yet we know that there is a lot of variability in the data, especially regarding the spectral distribution. So we wondered what was the intrinsic dimension of the data set. And for that, we try to estimate using different approaches and using a ground truth that we know specifically which was the dimension. And we end up by looking at the data could be a low dimensional organization between actually no more than 8 components, main components, to explain the variability. So we look for a method for visualization that could better preserve the local and the global structure of the data. And we choose the UMAP, Uniform Manifold Approximation and Projection, and it turns that reveals part of the ripple structure. We when we project the individual chart waves and the frequency of each of the chart waves into the two-dimensional or the three-dimensional manifold, what we noted was that the frequency of the events was remarkably well mapped in both dimensions. So somehow we suspected that the frequency was carrying information about the assembles, the microcircuit motif that were underlying the different forms of replay and the different replay events, from the slower to the faster components. Now, the two most important parameters in UMAP are the number of neighbors and the minimal distance, which are effectively used to control the balance between the local and the global structure in the final projection. So we try different combinations of parameters, and to our surprise, we found that the ripple frequency map was remarkably robust. So that it was quite clear that the frequency of the variability in the frequency of event is carrying strong information about the organization, or the underlying organization of the microcircuit that of this particular event. So to further convince ourselves that we have captured something true about the organization of the data, we derived a synthetic data set that had a similar distribution that the real data set. And what we found was that UMAP of these synthetic ripples mapped quite similarly from low to high frequency components, so that it seems that frequency should be one of those 8 dimensions somehow that we need to explain, that can explain how ripples is variable, so that we can hopefully model how the ripple frequency relates with the underlying circuit. But actually, identifying the different chart waveforms also works well with other methods based on spectral decomposition. And this is data from other paper using non-negative matrix factorization that they also found a diversity of chart wave ripples that could actually reveal differences between how the brain-wide dynamics evolve. In this very interesting paper by Ramirez-Villegas, they combine clustering of chart wave ripples recorded in the hippocampus of the monkey with brain-wide functional resonant magnetic imaging of the activity. And they found clear indication that the different ripple types somehow relate with activation of different circuits across the cortex and subcortical structures in the monkey brain, suggesting that somehow different interaction between ripples and neocortex contribute differently to how the reactivation pattern can actually scale brain-wide to brain-stained memories. And this is a possibility that, of course, suggests that you may be, by locally activated subset of ensembles activity in the hippocampus, you can actually broadcast differently through the whole brain and reactivate different, more extended networks of memory during these events. So actually, not only the different feed-forward and feedback microcircuit that wires the CA1, how they receive from the entorhinal cortex, from the CA3 and the dentagyros, how they are connected across the deep and these superficial cells within the CA1, and how these circuits are related with the activation order, either forward or reverse, matters. But there are also others aspects that could have a strong impact, like, for instance, different motivational states like anxiety, stress, novelty, the possibility to represent rewards. All these are associated to different modulatory components, or different modulatory pathways in the brain, which acts also brain-wide and influence how the plasticity operates and how the different circuits are active during a particular Charles-Ripple event. And that variability can hopefully also could reflect the other dimensions of the Charles-Ripples. Because indeed, replay is a very complex phenomenon, and most of what we know today about how the hippocampus self-played suggests we are just still on the infancy in trying to understand the many options that the hippocampus how they can play with memory representation and how to build memory. The most known role of ripples are in retrospective replay, which plays a role in memory consolidation by reactivating repeating patterns that were already experienced by the animals. Like, for instance, when the rat is in the location D, the cells, place cells from A to D are replayed either in the forward or in the reverse order, depending on whether the animal is sleeping or not. For instance, sleep replay is mainly forward, organized in the same order as experienced by the animal. In contrast, awake replay is mostly influenced by the animal location. And that is very interesting because that may suggest that somehow the place fields and the configuration of the entorhinal grid cells somehow bias the way in which CA1 sequences are replayed during awake situations. When the animals, instead of replaying the order from A to D, they reactivate memory traces or activate these sequences from D to A. That may suggest that actually awake replay may have a role in retrieving the information and informing directly the information which is available for the animal to make decision of where to go. But also in the most recent experimental results, using different tasks, having the animal moving around different type of mazes, we have learned that replay during Charles ripples, during awake Charles ripples, could be prospective, could be actually reflecting not only the sequence of events that happen, but the events to come. Somehow this prospective replay reflect the pathway ahead of the animal, and that is suggested that somehow it could be helping for planning. So it's this kind of organization in which the neural network can get information and can make a decision on whether to go in one direction or the other. But also we have seen that in some cases when the animals are exploring a room, but somehow we have not imposing particular trajectories in the room, the animal has a representation of the room, but not a representation of the sequence of trajectories that they can follow in the room. We have seen that in some cases a prospective form of replay, which is sequences that has not happened. I mean, sequence of events or sequence of place cells or sequence of location that have not been visited by the animals. And that suggests a kind of constructive role, a kind of imagination, something that could be somehow allowing to inform about the space of options that the animal may have. So somehow this is a big puzzle that we still don't understand, how a simple physiological event, which is Charles ripples, occurring during these different forms of replay, this different type of repetition and simulation of what is going on and how to make decisions and how to inform the brain to make a decision are occurring. And this is a fundamental questions right now in neuroscience and the main core of research in my lab and in other labs worldwide. So while AlphaGo and many other recent algorithms are now exploiting replay, mechanisms, we still need a better knowledge on how the many hippocampal solutions for self-training the network through repetition and simulation operates. And I have shown here how we can take advantage of some very simple machine learning tools to try to understand the brain that hopefully could also allow us to make wiser artificial intelligence solutions and to learn better how to implement more optimal artificial neural networks. And with this, I finish by thank you again, the organizer, for the possibility to talk about the research that we are doing in my lab in Madrid, and of course the funding agency and my group of enthusiastic young researchers who help in this endeavor on trying to understand how the hippocampus works and operates. And thank you very much for your attention, and I am happy to take questions.
Thanks a lot, Liset. We have around 300 people online, so I'll do the applauses. That's the applause for everybody else. And so we have a bunch of questions. We actually have at least right now 8. I'm going to share the screen with the questions, and I encourage everybody to vote right now. You can vote right now for what you think was the most important question. And let me just do the screen share. Let's see. Okay, so here we have the first question. And the question is, and you can still send questions right now, just in case you want. Joshua Benjo recently published that AI needs a prefrontal cortex in terms of the things that the PFC does, and AI fails to solve. What might be a special feature in biological learning that AI doesn't have currently?
Okay. I guess many things that there are that are not yet in the artificial. But yes, I mean, prefrontal cortex, this is a very important question. And for instance, during shardware ripples, some ripples, but not all of them, are associated with a reactivation of prefrontal cortical ensembles. So what prefrontal cortex, for instance, does is to have a top-down control of the sequences and the reactivation patterns. So, for instance, that level of hierarchies in the connectivity between the different brain regions and how they interact is something that we still need to distill better in models and in neural architectures to implement. So, as I said, somehow it seems that not all events are reactivated, not all self-play are run the same, not all of them involve these top-down influences. We don't know whether that could make a difference, but I beg my answer to the point that this is one of the directions that should be considered. Of course, I am not expert in artificial intelligence and machine learning and learning architecture, so probably there are early solutions that somehow implement this kind of hierarchies, but for sure this is one of them.
Thanks for the answer. One small request, Lisa. Could you turn off the screen share so that way, so we don't have the echo in the video?
Can I? What? What do you want me to do?
Just turn off the screen share right now, so that we can just share the screen with.
Okay, I'm sorry. Okay.
No. Thanks a lot. Yeah. So that was one of the questions. Let's go to the next most popular question. What type of new data types, experimental procedures are you most excited about for understanding the brain and interfacing with AI?
So for me, this interaction between machine learning tools and experimental data analysis, for instance, for a long period of time, basically what we do to understand electrophysiological events was running a spectral analysis. And we have known for many, many years that we are just not getting additional information from the spectrum. Surprisingly, the frequency is something which is informative. I talk about frequency in my talk, but there are other dimensions we are exploring as well. I didn't have the time to put them all. But the good point about these tools is that you can run them in an unsupervised manner and let the data structure to emerge, and then they show you things that you have not seen before. And then when you realize that there are these interactions between the data that you have not realized before, and you start as an expert to infer that information, this is extraordinarily interesting. And a good direction and a good new data types and experimental procedure that I think and we are more excited about is those with high throughput channel recordings like neuropixels that allow us to gather information from hundreds of channels, thousands of cells simultaneously, and that allow us to eventually evaluate more statistically the different sequences, for instance. And we are also pretty much excited about the multicellular calcium imaging, that while they are more slower in the time scale, not especially good for the chart wave ripples, they are extraordinarily good for other type of problems and looking at different organizations, like more at the behavioral scale.
Let's move on to the next question. Let's see. Okay, so this one's about one-shot learning. One-shot learning in the hippocampus, PFC, and brains seems to be a dream in actual AI systems. There's an actual theory that explains about how these areas accomplish that. And
Yeah, this is challenging both for artificial intelligence and for neuroscience. I mean, I just can speculate. So what we think is that, as I said, you can imagine that you have, how many sequences do you have in your brain? How many of them? How many of those sequences, meaningful, has been seen by you before? How many schemas are established in your brain so that you don't really need to train them too much just to recognize and to record them all? I do remember at some point, you learn what you already know. Learning new things takes a lot of time. But if, for instance, this is the case when you are looking for explanation, like, for instance, neuroscience, and you try to understand machine learning, use machine learning to understand the brain. This is fully new conceptual framework. So you don't have a schema for understanding that. You cannot do one shot, learning like, wow, this is UMAP, this means that. No, I can do that if I run a spectral analysis. Just this is a simple example. So one-shot learning, I think, is hard to get because it's require hardwiring and it requires some pre-existing networks there so that you can recall very quickly to those attractors and get informed about that. So, but this is a speculation, of course.
Let's go on to the next question. We actually have a lot of them. We have 8, and they keep on coming. Does the hippocampus only do positional maps memory, or does it somehow have to do something like cognitive maps in which we order a set of prior knowledge to solve specific tasks?
Of course. I mean, the hippocampus is a sequence generator. It's an associator. So space is just where things happen. But we know that when space is informative, the hippocampus will represent space. But if instead of a space, another input is informative, that will be represented by the hippocampus. And there are many papers showing that actually the hippocampal cell, they represent much more the association than the space per se. So, but space is a useful container of everything that happened as time. So whether it is a space or time or association between events that happen, whether the hippocampus just create that feelings of order, of sequence, is just fully open. So originally O'Keefe talked about the cognitive map, and the cognitive map is wider than just space. So where place cells were the way we start making ideas of how the hippocampus represent whatever happened, we know the hippocampus is important for episodic memory, for the narratives. That means the hippocampus is important for creating associations between what happened. So space is just where things happen.
So moving on to the next question, let's see. What is the status of the polychrony research line? Is there a machine learning interpretation of polychrony? Did I say that right? Polychrony?
Okay, I will Google polychrony. Polychrony, because it's the first time I heard about this. Polychrony is an integrated development environment and technology demonstrator. It provides model-driven environment to perform embedded system. So I have no idea of this. So I'm sorry about that. Is there? I'm sorry. I need to read a bit more about this. I'm sorry about this.
No. I mean, yeah, right. Gotta keep it honest. I don't know either. Let's go to the next one. Ivan Lozano asks, what is your opinion about the new OpenAI GPT-3 generating code from voice commands or text requests?
That's at the same level of polychrony. So I know that this GPT-3 is used for voice recognition, right? But I have no opinion about that. I'm sorry about that. I cannot tell.
No. I want to abuse being the moderator, and I actually have a question, and I think it's related a little bit to like the current world scenario. And so do you have any suggestions for example, for researchers that want to get into the field, or people that are just going to become professors right now, and we're entering into a time where there's like a lot of economic uncertainty. What do you think are areas where researchers can make an impact with like less resources? Because sometimes running experiments can be very intensive.
Yes, I know. Well, hopefully I think that, this is all about data analysis. But it's true that you need to understand and to interpret data. And but there is tons of data in there, and there are public repositories of data that can be used to build models and to eventually to analyze. But as I said, it is true that you need the interpretational background, so that you need an experimentalist somehow. But I feel myself that the field is evolving very quickly, and that data analysts of brain signals will generate a new interpretative field. So I don't think really you need, of course you need experiments, because that's the way to test predictions and to try to make sense of what you get. But you can also try to use data from very simple questions to address very simple questions, and there are these data repositories that you can have. For instance, we are working now in predictions of shareware ripples, occurrence of shareware ripples. For instance, we think that whether we will have a faster or a slower shareware ripple is something that we can predict based on what is going on in the few milliseconds, tens of milliseconds before the events is generated. And these are questions that could be useful for predicting seizures, forecasting seizures, looking at brain-wide interactions, EEG analysis. I think that it can be done somehow without experimental access.
Yeah, that's great to hear that you think there's a lot of questions you can still ask with like public data, and that just requires people to really like dig in with these new methodologies and tools, and maybe like new fresh questions. We're getting close to the end time, and we still have a few questions. What's the best way for people to, like, if people have other questions, do you have like a Twitter handle where, you know, people can be like, hey, Liset, what's if somebody has other questions relating to exactly your research, what's like the best way to interact?
To interact with me, send me an email. I mean, you can get in contact anytime, and I am happy to take questions and to interact with people around. And yeah, for sure. I mean, you can find me on Twitter or the website and send me an email, direct message, and I will be happy to answer the questions.
Well, thanks a lot, Liset. It was a pleasure to have you.
Thank you again for your invitation. Thank you for organizing this.
Yes, and we're going to move on to the next talk, which is from Eric Fala and IBM. So we're closing this.